If I could have each of y'all type in the chat just to say that you are consenting to the recording being on. That way everybody knows here. And I mentioned before I have uploaded the Word document that is the case study that we are going over. Let me see if I can pop that up on the screen here. So it is uploaded on, oh, come on, share screen. Okay, so it is showing, hopefully it's showing for everyone, the Word document there. It was under modules where you got to the link for this. It's just if you want to have that pulled up so you can keep track of cell Steve and cell Sutton and protein X and Y. I'm going to flip back over here to my presentation. Okay. So, I don't know if that is the chat showing up on y'all's screen from my screen, or is it not showing that? It's not supposed to be showing, but I don't know if it's showing it or if it's going to show my whole screen. But there we go.
There's your whole screen.
Okay. Alrighty. Well, we will just try to move that down. Okay. So for cell fusion, we are talking about two cells. They are cell Steve and cell Sutton. Originally, we were telling people like, okay, you have one cell from your retina and one cell from your pancreas, and people started getting confused on like looking up what the two cells were specifically making on for insulin versus rhodopsin and all these different things. And we were like, okay, no, just focus on general terms. And Steve and Sutton are one of the other professor's kids' names, so that's why they're named that. You can think of them cell A and B, whatever. It's just two cells taken from the same individual. So in the idea of you stole a cell off the back of one hand and a cell off the back of your other hand. They're an identical person, just two different cells from different parts of their body. Now, cell Steve makes two proteins, X and Y, and cell Sutton makes nothing. Yeah, it makes its normal cells to stay alive, but it doesn't make protein X or Y. So during our cell fusion, we are going to fuse them together, and you get a cell that has a mixed membrane and two separate nuclei still. Now, protein Y starts getting made by Sutton, and cell Steve stops making protein X. So the two questions there are, what is going to make one cell stop making a protein, and what is going to make another cell start making a protein? Or I think it actually says that just the mRNA. We won't even go through the full history of it, or the full pathway. But looking at it, I'm going to bring up now my whiteboard. Everybody clear on the idea, though, that we mixed the outer membranes, and now cell X, or cell Steve, is not making protein X anymore, and cell Sutton has now started making protein Y. So with that in mind, flip over to the whiteboard. Lovely. All right, clear. Why are you not clear? You're all drawings. And I'm going to be able to type on here. Okay. So I'm just kind of looking at, thinking about chapter 17 here, and we're gene regulation in eukaryotes. So we do have some extra steps that are going to occur for gene regulation in a eukaryote versus a prokaryote because of having the extra RNA splicing and the fact that it gives the idea that, make sure I say this correctly, it was, it says that protein X is a glycosylated trimeric protein. So that means that it's only, since prokaryotes do not usually have like the protein modifications and things like that going on, it's not going to be in a prokaryotic cell. We are working in eukaryotes. And because it says it came from multiple cells coming from the same organism, you, again, have to know it's a eukaryote. All right, it's back to my whiteboard. Okay, so our levels of regulation for gene expression, we can start with transcription. Translation. And we can fill in a few other things here for thinking about post-translational modifications, because we talked about it being glycosylated and a trimeric protein. I guess post-translational. We also get to have some extra stuff here with post-transcriptional. So in between, when we have turned it from an RNA, or we turned it from DNA into RNA before it gets turned into protein, and we can modify it here. Post-transcriptional modifications, translational modifications. Of course, somebody's going to call me now. No, not it. All right. Okay. So I'm just going to start off by listing out the ways that we can turn genes on and off. And then we're going to go back through and see like which ones would apply to this situation. So our first thing it ask, and y'all can turn on your microphones to chime in, or you can type on the chat as well as soon as I find where my chat screen went. Oh. All right, so easiest way, think back to like the lac operon. What did we do to turn off a gene? Or turn on a gene? Am I still coming through? Where'd my whiteboard go? Oh, you better not erase that. All right, well, I'm not hearing anybody talking. I see everybody is on mute except for Kelsey, but
I'm on.
okay, so looking at the stuff, really better not do this. Oh, I hate this program already. Yay, super. All right, so thinking about for transcription, we'll pop over here and see if we can change text color. All right. For transcription, we have the option that we can use repressors, right? Simple way to turn off a gene is repressor. Now, this is going to be something that it binds to silencers. We also have activators. And they bind to enhancers. You can also use co-activators. There is a step that you can change mediator binding, but that's not one that gets talked about very much. I'll put it on here just so we can compare it. And then there's some more on histones. So histone modification. So how densely you're packing your DNA to wrap around those histone cores. Now within this, you can turn it off or you can turn it on more. So methylation is going to silence it. It's going to decrease transcription. And acetylation is going to increase translation for that region. Whoa, okay, that was weird. Really? What the hell was that? This is making me mad. I'm just gonna start a new Word document or type it down on here. I can't believe it just did that. That makes me very mad. Looks like it just like erased everything that I did. Okay, yeah, great. It erased it all. I'm not liking whiteboard on Zoom, so we are just going to switch over, do it this way. All right. Okay, so transcription, post-transcription, translation, post-translation. All right, and over here we had repressors, into a silencer, activator, binds to enhancer. So methylation is decreasing transcription, and acetylation is increasing transcription. All right, now once we have turned it in from DNA, we went ahead, we turned it on, we turned it into RNA. What can we do to it before we turn it into protein? Now, since we said it was a eukaryote, it's going to have a 5' cap and a 3' poly A tail. So we can change our poly A tail length. We mentioned before that that was one way that kept it from being degraded. So if you were going to degrade something, you could change this, but 2 ways. There is this controlled by a polyadenylation signal sequence. And then you have enzymes to digest proteins. There's also splicing. You can have alternative splicing because it doesn't say that it turned off the gene. It just says it's no longer making protein X. It could be making a modified version. So just an alternative splicing. There are things that block splicing, so there's splice blockers and enhancers that change how this is done. You know, don't go into too much of the detail on it. And right at, we had just started getting into RNAi at the school. This is the inhibitory RNA that's going to bind to single-stranded RNA and make it trigger the degradation pathways. So RNAi is an option. Now, if we turned our RNA, we went ahead, we got it all the way to the ribosome, and now we can start changing how it's translated. Sometimes you end up with tRNA shortages. Not every cell uses the exact same number of tRNAs. So sometimes you'll end up with a slight variation even within a cell, like that the mitochondria use slightly different tRNA code than the rest of the nucleus, and it can sometimes be out of whack. So tRNA shortages, there's also the aminoacyl tRNA transferase or synthase. and amino acid shortages. You just straight up, when we talked about like feedback loops and how you, like, if we didn't go into too much detail on tryptophan, but just knowing that tryptophan existed. Dee Dee is here too. Dee Dee, if I can get you to just comment over in the chat that with the word yes that says you consent to this being recorded. The recording is running right now, so just letting you know that that is going on. The amino acid shortages, and then post-translational, we can have altered protein folding. And we said that this protein needs all 3 subunits. So if any of those aren't made properly, it's going to ruin the entire function of this 3 trimeric unit. Sorry, the trimeric unit is not going to form unless you have all 3 parts there. There's also the last step about protein function, that you can have degradation of the protein, and you can have feedback inhibition. So this is pretty similar to, I think it is figure 17.1 in the book, that it's the picture that shows all the different ways we can turn things on and turn things off for gene expression. Now, I want to go through and talk about cis-acting elements versus trans-acting elements. So cis-acting elements are things that are acting on self, and trans are things that can be moved around. Like the repressor for the lac operon is floating around. That would be a trans-acting element. So I can type in here, the repressor can move around, but any DNA sequence can't move. It's considered a cis element. Same thing that an activator is a trans-acting element. It floats around, but the enhancer sequence is stuck in place. Mediators float around. They can move. Now, the histones themselves, like once they're put into place, are cis-acting, but the enzymes that do this, so the enzymes causing these changes are transacting. Like your kinases and stuff. Now your poly A tail length, the poly A signal sequence can't move. That is already encoded by the gene. It's cis-acting. But the enzymes are transacting. Splicing, I guess those would be considered transacting, because they can move around and change. RNAi is trans. And pretty much anything once you get down from this area forward is going to be transacting, because it's you can move the protein around, or you can move the RNA around, so it can move to whatever needs to happen. All right, everybody okay so far? Anything y'all want me to go over in more detail before I keep going? Here's my chat screen.
No, we're good.
Okay. All right. I lost my screen. Okay. So thinking back to our picture of our proteins here, we have This is going to show my screen. All right. So our question tells us that protein X has stopped being made by Steve. So we want to look at our list of things and say, what would make protein X disappear or stop being produced? And then we will come back and look at why protein Y's mRNA being made. So the first thing I want to point out is, do these cells have the same DNA sequence in them? Now, we said as part of our instructions here that they came from the same individual. Up here. You experimentally fuse cells Steve and Sutton, which are from the same organism. So that means they have the same DNA code, the same DNA blueprint inside both of the cells. That means we can rule out any changes that were occurring due to mutations or changes in DNA sequences. When we fuse these together, they are sharing their signals, but their main sequences are going to be set. So we want to find things that are now able to be shared across their nuclei. They each have their own membrane there, but things that can cross over from one cell to the other. And of course, if we're going to be turning off a cell, the first one we can look at is repressor. Turn on my highlighter. So a repressor, that was already in place in cell Sutton, can now float across and bind to the DNA silencer sequence in cell Steve's DNA. Does that make sense? So if I, let's see, it's going to be a really terrible picture here. But okay. So before, here is the nucleus from Steve, and it's got his DNA in here. Here is Sutton, and here is his DNA. Now, normally, we would get protein X being made here. This is gene X. But over here, we don't get gene Y because let's say that there was a repressor sitting here. Now, this repressor is going to stop transcription from happening. Now, when we fused our cells together inside one big membrane, the repressors that were floating around inside here could have actually, what would have probably been floating around inside the cell. Now, when you fused them together, one of those repressors can come bind over here, and now it's going to knock out that signal, or it's going to knock out the transcription of that mRNA. Make sense? So when I'm asking over in here, hopefully this is actually, draw a diagram to explain the results of the cell fusion experiment. Add numbers that correspond to the pathway points mentioned in your first answer. If you had these numbered, you can draw a very, very simple picture like my terrible sketch here. You can use it, or you could do a Paint or PowerPoint or however you want to do it instead. I don't care how you do it, but this is what I mean, is you could just, even if you zoomed in only to this spot and said a repressor from cell Sutton is now binding to the silencer sequence in cell Steve to turn off gene X. That's all we need to do. So now I'm going to show another option here. clear the drawing. And the next one was the activator. Now, something that could be happening that's a little bit different. Oh, look, I can do it this way. Here is cell Steve. Here is cell Sutton. And here is our big one there. Now let's say instead, this gene only gets turned on when we have an activator here. And over in our other cell, it doesn't have an activator. So that's why this one doesn't normally make the protein X. It's missing its activator. Now, let's say that there is a signal that comes in and somehow is degrading this activator. We have some different versions. If we go back, I'm trying to find out what page in the book it is on. This was from last week. We talked about inducible proteins, things like that. So if you had, you can sometimes have the activator that needs to have an inducer to bind. That's what was happening in the lac operon with CAMP and CAP. Or you could now have an inhibitor protein. Let's say there's little inhibitor proteins floating here. And now one of those inhibitors is coming over and binding, and now that's causing it to no longer be able to bind. So the activator is being shut off by an inhibitor, keeping it from binding to the DNA, so it can no longer be turned on. All right. So that gives us two options there. So we saw that the DNA sequence didn't need to change. We didn't add a silencer sequence. We didn't add an enhancer. Those were already there. We are just changing these transient signals that can go around. Histone modification is something that's easier to talk about too. So in this case, we've talked about X and activation previously, and we said if you added methyl groups to a gene's histones, you could no longer untangle that DNA from the histone. It's got a charge that's going to make it stay more tightly bound. And if that promoter region is wrapped up on a histone, we are not going to be able to turn on or have anything binding to the promoter region. So methylation is going to keep a gene silenced. And this is something that can be turned on and off during the life of a cell. A lot of your methyl groups get set down during early prenatal development, but exposure throughout your life to different chemicals can change these pathways as well. That's sometimes what happens when cancer gets turned on. It changes methylation patterns. English. All right, so that's just ways that we can turn off transcription. And if you, again, are looking at figure 17.1 in your book, regulatory transcription factors can activate or inhibit transcription. The arrangements and compositions of nucleosomes, those are your histones with the DNA wrapped around it, influence transcription of DNA methylation, usually inhibits transcription. So for our RNA processing, according to the book, so this is post-transcription, pre-translation. The first one I have listed here is about the poly-A tail. Now, our poly-A signal sequence isn't going to change. So that's something that we can say, no, won't change. But the enzymes to digest that poly-A signal could change. When you when you fuse the two cells together, you could have whiteboard. If these were enzymes instead that were floating around here, we can make some a different color. So let's say these are our, oh, here, I'll do this. This is Pac-Man. He goes chomp, chomp, chomp. So we wrote our mRNA. It's a slightly different color now. Here's our mRNA, and here is its poly A signal. Let's say when we fuse these together, Pac-Man can now come over and chomp on here and degrades this RNA before it ever gets turned into protein. Now, most of those signals to digest would have probably been in the cell already. So I'm gonna actually mark this one as orange. It's kind of it won't let me. We'll do pink. It would be unlikely to have changed because the enzymes to digest the poly A tails are not gene specific. They're going to chew up any poly A signal. So if you weren't degrading your mRNAs before, you probably wouldn't have survived. You do need to be able to turn those signals off. So while it is possible, that's not going to be a heavy influence on this one. But alternative splicing, and because we said this was a trimeric protein, depending if it's an operon where all three are transcribed at the same time, that is something that could change. There are some different blockers and enhancers that could go on. So changing splicing patterns, it could be still writing down the DNA into mRNA, but then that mRNA is being read differently. And RNAi is another thing that can totally happen here, because if we go back to my whiteboard screen. So instead, if I take off this guy, and go back to alignment. Here is our mRNA, and it is single-stranded. You can have small interfering RNAs going around over here. So I don't know if you can see the difference. These are red. And if that sequence matches here, this is going to trigger the risk pathway. So this is RNAi, inhibitory RNA. And whenever you get double-stranded DNA or nucleic acids in general outside of the nucleus, that signals that this could be invading viral DNA, and the risk pathway is going to grab a hold of it. And that is going to signal to degrade this mRNA before it gets turned into a protein. This is, again, it's kind of like the immune system inside a cell. All right, now flipping back over. Okay, so we have splicing. Now, it is possible that you could have these types of amino acid shortages and stuff, but given that these cells came from within the same organism, it would be fairly unlikely that that would be causing the issue, because if one cell has the appropriate amino acids, the other cells would probably have their appropriate amino acids too. So I'm going to flag this line again with that like kind of pink color to show that while it is possible, it's probably not what would be going on here. Now let's say the mRNA did get turned into its polypeptide signal or sequence, but you could have altered protein folding. So looking back here at my whiteboard. And I will erase this guy. He's gone. Okay. All right. mRNA gets made, our ribosome attaches to it, and out pops kind of a protein. You sometimes get these other things, chaperone proteins, and they actually work kind of like a barrel, the lid on it. And this new polypeptide, as it comes out, goes into the barrel, and the chaperone helps fold it up so that when it comes out the other side, you have like a very specific fold. We're going to pretend that's what our protein looks like. So if something happens to change the chaperone protein that is normally folding, some signal coming in, and now this chaperone from cells Sutton is modified in some way, that now it's making a protein that looks like this. So instead of having like a square slot there, it's got a triangle. Now this is no longer going to be able to fuse where it needs to. So this is a non-functional protein. Non-functional. So alternate folding will screw up your cell. And I mentioned also that it was a trimeric protein. So you have 3 different parts to it. It's going to give you lots of 3 different ways to break it. So if any of these subunits get changed, now you don't get a functional protein. And in terms of protein function, if you had something that was some sort of enzyme, again, Pac-Man style, coming up and chewing up your protein, the fact that it said that it never made it to protein form, these things aren't going to apply to the question asked. So these are the ways that we can repress a protein expression. Now, the other question on here is nucleus derived from the cell Sutton is producing mRNA for protein Y. Okay, so now we want to look back through here. I'm going to change off our colors. Erase our highlighters, marks, and talk about ways to turn on a cell, or turn on a gene. So if we had a repressor that was shutting something down before, we can go back and look at our whiteboard. I'm going to erase this stuff. Because, oh no, I'll put the cell back. I see Dee Dee's question here. Yes, yeah, it's, well, altered protein function versus like closed conformation, depending on what you mean by closed conformation, in terms of protein structure, yes, anything that's changing the structure of a protein. So if you've ever seen like the pictures of an enzyme where the inhibitor is in it and it's binding and changing it from allosteric regulation points, any of those things, like a protein, its 3D shape is its ability to bind to stuff and do its job. So if anything is changing the shape of the protein, it's no longer going to work. Whether that's the chaperone isn't there at all, its chaperone got degraded, and so it no longer gets folded up into a nice, neat shape, or that when it folded into the wrong shape. Any of those changes to it is going to cause it to no longer function in the way it's supposed to. So yeah. All right. Hopefully this is sharing to the right one. All right. Adding back in my purple here. Here is. All right. So Sutton's back. Now Sutton has started making Y when it didn't before. So we had our option, let's say before there was a repressor sitting here. So I'm going to remind y'all back with like the lac operon and how repressor sat there until allolactose came in. So now suddenly we have some sort of signals coming from when these were bound or when we fuse the cells together. Now something comes and binds to this repressor and it falls off. It's gone. I will. Or maybe the repressor is still there, but it is bound to an inducer here. And so it can no longer bind, and we're going to end up getting this gene now turned on. This is for gene Y. So changing the repressor by having something be able to bind to said repressor, in this case, that's called an inducer. An inducer coming in and binding is going to make the repressor fall off. This is just like what happens in the lac operon. We also have the idea, again, of activators. So an activator could come in from over on the green side, and this activator now pops on, and that's going to turn on. That's probably the most likely way, but not the only way. So a repressor could be changed. An activator could certainly now happen. Again, mediator, that's going to work in conjunction with the other signals. So you don't have to necessarily worry about that at this point. It's usually that the silencer sequence and the enhancer sequences have to get bent around via a mediator. And if your mediator's gone, it would mess things up. But now histone, if they had been silenced due to histone methylation before, histone acetylation could turn them on. So now that would change and would open up the promoter regions or the enhancer regions or whatever regions that were being blocked before by the methylation can now be open. And the question just says that it's producing mRNA for protein Y. It doesn't go through to say that that protein is getting turned into a full protein. So we only have to talk about transcription on the first part because that's as far as the question asks you to go. All right, let's sure. So, are there any questions? Do those ideas make sense? Y'all know the figures that I talked about in the book. Figure 17.1, it gives just all of the general steps for where these things can be turned on and turned off. And also, just to remind you, back at figure 16.2, where it talked about activator proteins and repressor proteins and inducers and co-repressors and all of those kinds of things. Because there is the other idea that maybe the repressor protein was already floating around inside cell Steve, and when they fused together, cell Sutton just gave it a co-repressor. So now that repressor can bind. All of those are separate answers that can happen, just to give you more jumping off points. Because I'd like to have y'all show me at least a couple versions of how we can turn things on and how we can turn things off. Remember, you don't want to do anything that is going to change the DNA sequence because the DNA is going to be the same for both cells. So that won't change it. You're not going to fuse two cells and suddenly have a new protein unless you messed up when you did yourself. Or you suddenly have a different DNA sequence unless you screwed up your fusion process, in which case you don't want to do that. That's not what's happening here. So figure 17.1 and figure 16.2, and your pictures can be as simple as the ones I drew. You can draw them by hand and take the picture and upload it, or you can draw it on Paint or whatever photo program you want to use. Not being judged on your artwork, you're being judged on the idea behind the art, that you understand what things are going to turn things off and what are going to turn things on, and that you understand where they came from. All right, so does anybody have any other questions? Anything you want me to talk about? Hopefully the screen shares worked properly when I was switching screens and everything was viewable on the video. So you can go back and rewatch this anytime you want. Again, question 1 is for turning off protein X. Question 2 is for turning on protein Y. And so if you list them off in order, I'd say give me a couple examples. Probably, yeah, I mean, you have a lot more options for number 1. Number 2, you can give me, like, I guess there's 3 main ways, but there are actually more. I think we wrote it out. There were like 11 for number 1 or something like that. I don't know, because all of my paperwork's still sitting on my desk at school that I can't access. But so we'll say, and I'll include this on instructions, to give me 5 ways for question 1 and 3 ways for question 2 for the figures. And since I already went through and gave y'all all of the answers, it shouldn't be too hard for you to come up with them. Just draw me little pictures for them. All right. I'm going to go ahead then and stop recording here.